Good morning. It's August 20th, and this is your daily brief in science. Here's everything you need to know. A new large analysis links leisure time movement to lower dementia risk, while work-related activity shows the opposite signal in a surprising twist on how movement affects the brain. Researchers pooled data from more than 4.2 million participants across 30 countries, tracking dementia outcomes over a median follow-up of 10 years. The study found that leisure activities such as walking, cycling, running, swimming, and sports were associated with a significantly reduced risk of all-cause dementia, including Alzheimer's and vascular dementia. In contrast, occupational physical activity was linked to about a 20% higher dementia risk, with active commuting showing a modest increase and household activity remaining inconclusive due to limited data. The authors emphasize that context matters. The total amount of movement can have different brain health effects depending on whether it is self-directed, leisure, or work-related exertion. Occupational activity often involves repetitive, low-complexity tasks, exposure to air and noise pollution, fatigue, and psychosocial stress, which may limit cognitive reserve. Leisure activity tends to be self-directed, varied, and socially engaging, providing opportunities for stress relief and cognitive stimulation. Editorialists from University College London note that occupational activity can coincide with factors such as lower education, less cognitively stimulating work, exposure to neurotoxic agents, and higher smoking rates, which could contribute to the higher observed risk. This represents the first systematic examination of the dementia-specific physical activity paradox, showing that non-pleasurable work-related movement may not yield the same brain benefits as voluntary exercise. The study, published in The Lancet Public Health, calls for public health strategies that improve access to recreational spaces and reduce harmful workplace exposures to maximize brain health. Researchers caution about reverse causation in observational studies, where preclinical dementia could reduce leisure activity and exaggerate protective associations. A phage-encoded kinase called T7K can disable bacterial defense systems that target nucleic acids by phosphorylating key proteins, as shown with RetroNECO9 and the DARTG1 toxin-antitoxin system, where phosphomimetic changes abolish defense activity. T7K specifically targets DNA-binding proteins, enabling it to undermine nucleic acid-targeting defenses across multiple systems. Phosphoproteomics during infection revealed extensive remodeling of host and phage proteins, detecting over 15,000 phosphopeptides and showing most depend on an active T7K. The study identifies phosphosites on RetroNECO9's RCAT and on DART within DARTG1, with phosphomimetic substitutions compromising defense, indicating phosphorylation reduces toxin activity or stability and neutralizes anti-phage responses. T7K has a broad substrate range, phosphorylating serine, threonine, and tyrosine without a strict sequence preference, biased toward nucleic acid-binding proteins because of its DNA-binding C-terminal domain. The kinase phosphorylates numerous host proteins at high stoichiometry, including the transcriptional regulator RCSB, linking to silencing of host defenses and aiding phage replication. Deletion of the SO domain reduces overall phosphorylation by about 31% and shifts the phosphorylation landscape, suggesting the SO domain targets substrates and sustains kinase activity. Phosphorylation follows a rapid wave within the first 5 minutes of infection, peaking before autoinhibition and shaping early host-phage interaction dynamics. This finding frames T7K as a broad countermeasure against bacterial defense systems, highlighting a new mechanism in phage bacteria immune evasion and mapping the phosphorylation landscape that accompanies infection. The article cites licensing details, CC by 4.0, for expert opinion and figures, and provides a DOI to the Nature paper, Bartolek et al., Pervasive Phosphorylation by Phage T7 Kinase Disarms Bacterial Defenses, Nature 2026. A growing population of humpback whales is frequenting New York's coastal waters, drawing public excitement and prompting policy attention to protect them amid heavy maritime traffic. Whale sightings have become common thanks to cleaner habitats from environmental protections, abundant prey like Atlantic menhaden, and climate-driven changes in prey distribution. NOAA designated an unusual mortality event after the 2017 to 2018 period following delisting as endangered, due to risks from vessel strikes and entanglements in busy ports. Local experts say mariners need new skills to coexist with whales, including recognizing whale behavior and reporting sightings to prevent strikes. Notable behaviors observed include lunge feeding and breaching, with individuals such as NYC-0318 becoming local favorites for spectators. The Whale Awareness Act, signed by Governor Kathy Hochul, integrates marine mammal protection into state boater safety courses and vessel registrations to educate mariners and reduce collisions. The Whale Cognitive Act requires marine mammal protection content in safety training for ship operators and in vehicle registration processes, reflecting policy responses to risks. New policies advancing risk mitigation include the Whale Awareness Act, which embeds marine mammal protection education into state boater safety and vessel registration. Despite the risks, the conservation narrative highlights whales as a growing hallmark of New York's coastal environment and boating culture. Educational initiatives for seafarers include an online course by Carl Loebu and the Nature Conservancy focusing on whale behavior, sighting reporting, and collision avoidance. Ocean expert Carl Loebu is developing guidance materials to help crews recognize whales, report sightings, and navigate among whale populations. Researchers and conservationists are leading educational efforts, using online courses to reduce collisions and improve safety for whales and humans. A new imaging breakthrough could reshape how we study biology at the smallest scales. Mosaic is a reconfigurable multimodal optical microscope that combines more than 10 imaging techniques into one compact instrument, allowing rapid switching between modes to reduce sample perturbation and preserve biological context. Adaptive optics and a modular switching system let Mosaic move between imaging modes in as little as 2 to 5 seconds, cutting blur from living tissue and enabling high-throughput data collection. In practice, the platform cycles through about a dozen imaging modes in seconds, using a shared set of lasers, mirrors, cameras, and computing hardware to deliver sharp, high-density data with sample-induced blur corrected. Berkeley Labs' LDRD-supported efforts, including the Pedikit 5D software, enable real-time handling of terabyte-scale data and aim to train AI models that reason over 5-dimensional data, 3 spatial dimensions, time, and molecular identity. Mosaic can generate up to 4 terabytes of data per hour, underscoring the need for advanced computational workflows, real-time processing, and large-scale data analysis to extract biological insights. The broader vision envisions self-driving biological laboratories, where automated microscopes, sample handling, and perturbation systems accelerate discoveries by integrating experimental context with AI-driven analysis. Future AI capabilities under development include vision-language models that reason over biology to guide experiments and potentially enable autonomous laboratories. The team notes that the bottleneck has shifted from data acquisition to turning dense, high-dimensional observations into biological understanding, relying on computing, AI, and automated instrumentation. VIPS used Mosaic and computational tools to image 2 mouse olfactory bulbs at nanoscale, generating roughly a petabyte of data in about 2 weeks, illustrating the processing gap between acquisition and interpretation. The VIPS project showcased volumetric imaging via photochemical sectioning, imaging 2 complete adult mouse olfactory bulbs at nanoscale and highlighting the data handling challenges involved. Mosaic has been demonstrated across diverse biological contexts, including tracking single molecules in living cells, mapping neuronal architecture in human brain tissue from an Alzheimer's patient, and imaging neural activity in live mice with adaptive optics, increasing detectable calcium events by about 2.5 times. A new effort to translate brain imaging findings from rodents to humans is underway, aiming to add under a minute to standard MRI exams by running parallel rodent and human magnetic resonance elastography studies in in-building imaging facilities. Researchers plan to adapt the approach for humans while accounting for species differences and practical clinical needs, such as speeding the scan and weaving MRE into routine MRI workflows. The project emphasizes concurrent imaging in rodents and humans to accelerate translation and make MRE a common part of brain scans without substantially extending visit times. Published in Brain Communications, the study reports estrogen-related, region-specific brain adaptations across the reproductive cycle and seeks to extend these observations to people. UD's in-building human and animal imaging facilities are highlighted as a key advantage for cross-species collaboration and faster translational progress. In the rat, MRE revealed estrogen-dependent mechanical changes in the hippocampus, establishing groundwork for how hormonal shifts may affect brain health across life stages. Future work will look at mechanical changes during menopause, testing whether estrogen depletion could create a mechanical, stuck state that might relate to cognitive symptoms such as brain fog in menopause models. The overarching goal is to present brain mechanics as a complementary readout alongside chemistry and circuitry to understand hormone-driven brain remodeling across reproductive life stages. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.